Okay, so today we're going to do something a little bit different from the presentations that you typically hear on the call, which Jenny just reviewed for you. But we're going to take a step back and talk about ClinGen as a whole. And what I'm going to try to do is provide you with a high-level overview of the accomplishments that we've made over the past almost 10 years. And I hope you walk away with a better sense of how all of the various ClinGen activities fit together and how your work is contributing to the resources that we are building. So, as you may know, ClinGen was initially funded primarily by NHGRI in 2013, and our current funding is set to go through 2026. The program has always represented a strong collaboration between 3 separate grants, though the composition of each grant has changed over time. Today, the 3 grant teams are Baylor-Stanford with co-funding from the National Cancer Institute, and the PIs there are Sharon Plon, Alex Milosevic, and Terry Klein. The Broad-Geisinger team, where the PIs are Heidi Ream, Krista Martin, and myself, and UNC and Company, where the PI is Jonathan Berg. And ClinGen is governed by a steering committee that includes all 7 PIs and additional key representatives from each grant, as well as NHGRI and ClinVar. We have, and I actually didn't even update this, we have even more than 37 working groups and over 100 different expert panels. And we've got over 2,100 people that contribute to ClinGen from 59 different countries. So that's who we are today in December of 2022, but how did we get here? So how did ClinGen even come to be in the first place? So ClinGen was really born from a need to develop resources to support the genomics community as they continuously work to understand the roles particular genes and variants play in human diseases. And over time, we've all watched technologies evolve. As we get better and better at detecting variants, the more likely we are to find ourselves in situations where we're evaluating something that may never have been seen before. And this situation is not just unique today, but it's in fact been happening for many, many years with each incremental step forward in technology. But really around the time when microarrays and the next generation sequencing technologies came into wide clinical use, so think like early 2000s, 2010s, people really began to organize around the need to work together, to set standards for how variants were going to be evaluated, to identify the types of evidence needed to support those evaluations, and then to ensure that people could easily access that evidence. So when thinking about the types of evidence necessary to interpret a variant, one of the first things you probably think about is just information about other observations of the variant. And at the time, most variants from clinical laboratories were not being shared publicly. Really, the only experience you would have to draw from was often from your own lab, which was likely quite limited. And clinical labs were doing redundant curation work, and they were often not coming to the same conclusions, partly due to unequal access to evidence. And this issue was observed in both the sequence and the cytogenetic variant communities. The cyto community was one of the earliest to mobilize on this issue, establishing the International Standards for Cytogenomic Arrays, or ISCA consortium in 2007, and then working to establish procedures through which labs could share variants that they were identifying on microarray through dbGaP and dbVar at the time. And though data ultimately was shared through these venues, the process was really difficult and the data was often not easy for others to find or access. So by 2011, a group of laboratories, having reviewed and ruled out numerous other potential solutions for a variety of reasons, including cost, issues with trust, accessibility, stability, etc., approached NCBI about building a variant knowledge base. Incidentally, NCBI had already begun planning for such a database for clinical variants as an expansion of dbSNP and to support the genetic testing registry. NCBI obviously has a reputation of building and sustaining valuable resources, and that kind of obviates the issue of commercial bias or conflicts of interest. So at that point in time, the clinical laboratories and NCBI began to work together. And this, as you likely have guessed by now, was the beginning of ClinVar. So from that point on, what we now know as ClinGen and ClinVar began to evolve together. The labs, including the ISDA consortium and sequencing laboratories, were trying to secure funding of their own to continue to support their work in data sharing and building variant interpretation standards. Meanwhile, NHGRI put out an RFA looking for groups to help develop a resource to identify clinically relevant variants, to which the Stanford, Baylor, and UNC teams applied. Late in 2012, NCBI launched a prototype of ClinVar, including early submissions from ISCA and the collaborating labs. And then in 2013, ClinVar was officially launched, and a few months later, all three ClinGen grants were funded. And from that point forward, ClinGen and ClinVar have worked in concert with each other. So one of the absolute most common questions that we get is, what is the difference between ClinGen and ClinVar? Why do we need both of those things? So for starters, and I hope I can convey this to you through the rest of the presentation, these are both monumental tasks that could not effectively be done by one group alone. ClinVar, of course, serves as a public archive of reports of variants and their relationships to human disease. They are a submitter-driven resource that's charged with cataloging, aggregating, and maintaining the millions of variant submissions that they receive, and then presenting that information back to us through their public user interface, their downloads, etc. Because of their role as an archive, ClinVar is not passing judgments on any of the submissions they receive. They don't classify the variants themselves. They don't assess the validity of any claims made. And they, by and large, accept most of what they receive, with rare exception. Without them, our community would not have access to the volume of data that we have access to today. Now, of course, they are sustained by intramural NIH funding, and they're maintained by the National Centers for Biotechnology Information. And because of this, it has remained relatively stable, as it has over the past almost 10 years, and available for the foreseeable future. There aren't any issues with commercial ownership or competitive grant renewals here, which were key considerations when thinking of how to best store this type of data for the long run. ClinGen, on the other hand, is supported by grant funding by the NIH. And one of our mandates is to encourage data submission to ClinVar. And in the past, we've incentivized ClinVar submission, we've assisted groups as they work through the process, and we have directly facilitated certain submission, such as those for patients, which I will get to later. Once this type of information is publicly available, then ClinGen can use it to answer critical curation questions about genes, about variants, and then provide that information back to the public through other avenues like our own website. We are also continuously providing feedback to ClinVar on things like usability, improvement requests, etc. But ultimately, both groups are working together to improve our knowledge of genomic variation. So I'll just spend a moment reviewing how ClinVar has grown over the past 9 years, and then we'll spend the rest of our time discussing ClinGen's growth. But as I mentioned, ClinVar officially launched in April 2013, and at launch, they had about 28,000 submissions from places like OMIM, the ISCA consortium, and those other partner laboratories that we talked about, so about 20 unique submitters in total. Now, at the end of November, so just last week, there were over 2.4 million submissions from over 2,300 submitters around the world. And in this graph, you can see the number of submissions growing each year in the blue bars, on average about 76% per year. And the orange line represents the increasing number of submitters, so on average about 96% growth per year. And this is really a testament to how the community has embraced data sharing and recognized it as a key aspect of variant evaluation and laboratory quality assurance. So now on to ClinGen. When we first started in 2013, our mission was to build and support authoritative central resources that define the clinical relevance of genes and variants for use in precision medicine and research. So how have we done? I'll use this infographic to kind of walk you through our approach to this mission, which begins by aggregating genomic and health data from various partners. And the sharing and aggregating of genomic data is, of course, critical to our mission. As we've discussed, this information is the evidence that we need to move forward with our evaluations of genes and variants. If people don't have access to the same pieces of evidence, then they may come to different conclusions in their gene and variant interpretations. And this can lead to mistakes in patient care. So one of our key efforts is to encourage data sharing and provide tools to aggregate such information for public use. I do think it's important to point out that ClinGen itself does not generate any new sequencing data. Our work relies heavily upon aggregating previously generated data from multiple sources. And this includes things like classified variant and phenotype information in ClinVar, but also things like population data sets, so think GNOME, existing gene and variant annotations, the published literature, et cetera. All this information can be either programmatically or manually annotated in our curation interfaces for downstream use as we answer our critical curation questions. We've already talked about the fact that ClinGen encourages and facilitates submissions to ClinVar, but I did just want to highlight the work that we do to facilitate submissions directly from patients. So we created Genome Connect in 2014 as a way to engage patient partners in data sharing, allowing them to contribute both variant and phenotype data to ClinVar in a de-identified manner. Genome Connect is open to anyone who has had genetic testing, regardless of results or diagnosis, so it is not gene or disease specific. People enroll online, they consent to de-identify data sharing and recontact, and then they provide their health history through online surveys and upload a copy of their genetic test results. We currently have over 7,000 people consented for data sharing, either through Genome Connect or through our other patient registry partners. And we've submitted over 3,500 variants to ClinVar on their behalf. Though when we initially developed Genome Connect, we were thinking it was going to be a way to increase the amount of phenotype data available within ClinVar. We actually find that almost half of our submissions actually represent novel variants that have not previously been submitted to ClinVar. And so without these patient partners taking the time to donate this information to us, these variants would not otherwise be publicly available. Again, ClinVar's focus is really to serve as a repository for variants and assertions about their impact on human health. But ClinGen, of course, has tools that support variant evaluation in different ways. So I'll point out the ClinGen allele registry, which aggregates variants from multiple sources, including ClinVar, and provides stable, unique identifiers for those variants. And having such canonical identifiers available is crucial for exchanging and linking information about variants across different data sources, particularly given the number of ways to correctly describe the same variant, even if you're strictly following HGVS nomenclature standards. The number of variants registered in this resource has increased to almost 2.6 billion. On the other side of the screen, we have the linked data hub, which is a service that leverages those unique identifiers to aggregate annotation information, such as molecular consequence, allele frequency, in silico prediction source, et cetera, on particular variants and facilitate the variant prioritization and curation process. And currently, the linked data hub has data on over 9 million variants and 119 genes. So going back to our infographic, once data has been shared and aggregated, we can use it to answer our curation questions as shown in the middle. So I'll walk you through each of ClinGen's current major curation activities to give you a better sense of the types of evaluations that we've been doing. And we'll actually go in order of when the curation activities were first developed. So first we'll talk about dosage sensitivity. And dosage curation first started as an activity within the ISAA consortium that we were talking about earlier in 2011 and came under the ClinGen umbrella as a result of the two groups merging. So here, the curation question that we are trying to answer is, does loss or haploinsufficiency or gain, triplo sensitivity of an entire copy of a gene or a region cause disease? And this information is different from predictors of haploinsufficiency that you might be familiar with, such as the Decipher Haploinsufficiency Index or the NOMAD, PLI, or LUF scores, as the final classifications are determined based on observations of affected human individuals. This information can be used to aid in the classification of constitutional copy number variants and has been recognized as an important piece of evidence in the professional guidelines for those types of variants. Dosage information can also be used when trying to determine whether haploinsufficiency or triplo sensitivity are valid mechanisms for disease. And as of earlier this month, the dosage group has evaluated over 1,500 individual genes and 500 genomic regions. So this graph is just showing you the growth of dosage sensitivity curations over time. You'll notice a few spikes in progress, and those correspond to concerted efforts to auto-classify, for lack of a better word, straightforward genes or regions, such as those observed at high frequencies in the general population. Dosage currently has 3 active subgroups focused on the evaluation of recurrent regions, such as those mediated by segmental duplications, as well as single genes involved in neurodevelopmental disorders and hereditary cancers. At this time, about 65 people curate for ClinGen dosage sensitivity. We'll do this for each of the major curation activities, but to quickly walk you through some dosage milestones over the past several years. Again, our group was convened in 2011. Our scoring framework was published in 2012. By 2018, we were able to publish a paper describing how ClinGen dosage information could be used to resolve CMV discrepancies in ClinVar. In 2019, we updated our scoring framework to be more stringent and more consistent with other subsequent ClinGen curation frameworks, such as the one for gene disease validity. And then for years, our curation data was publicly available on a separate site, again, stemming from that history as a separate organization. But in 2020, all dosage data became available on clinicalgenome.org, resulting in a large, sustained spike in web traffic. Also in 2020, our curation data was recognized again as a vital piece of evidence in the constitutional CMV evaluation process. And as part of this recognition, we undertook a project in 2021 to bolster the regions curated as dosage sensitivity unlikely and therefore be classified as benign by leveraging population data from NOMAD-SV and the database of genomic variants. Moving on to actionability, here the curation question is, which genes, when significantly altered, confer a high risk of serious disease that could be prevented or mitigated if the risks were known? The actionability team uses a semi-quantitative metric to assess outcome intervention pairs based on the severity of outcome, the likelihood of disease, the effectiveness of the intervention, and the burden or risk of the intervention to the patient. This type of information can be used to guide decisions on reporting secondary findings and is considered by ACMG when updating their list that they recommend labs routinely assess for secondary findings. And as of earlier this week, a total of 174 actionability reports are available encompassing 254 different genes. So actionability curations have been available since 2014. There are currently 3 active subgroups focused on adult and pediatric actionability, as well as a newer group focused on the adaptation of actionability metrics for the evaluation of polygenic risk scores. And there are currently about 39 members of our actionability team. Notable moments in actionability history include the publication of their initial adult-focused framework in 2016. Then in 2017, the ACMG Secondary Findings Committee kind of officially acknowledged that the ClinGen actionability reports were an important part of their evaluation process as they worked through new nominations to the secondary findings lists. In 2018, the group expanded their scope and began adapting their framework in order to be able to evaluate actionability of secondary findings observed in infants, children, and adolescents. And in 2019, the group started work on translating their actionability scores into assertions. So think like definitive actionability, strong actionability, moderate actionability, et cetera, in order to more easily convey their findings to users. In 2021, the group expanded their scope again and began considering how the existing framework could be adapted to assess the actionability of PRS scores. And recently, just this year, the pediatric framework was published. So next, we'll discuss gene disease validity. Here, the curation question is, does pathogenic variation in a given gene cause disease? Now, many claims about genes and their relationships to human disease have been made over time, but which ones of those are actually valid? So to address this, ClinGen has developed another semi-quantitative metric to describe the strength of evidence supporting or refuting such claims. And this type of information could be used, for example, by a clinical genetic testing laboratory when building tests for certain indications or when developing analysis pipelines. As of the end of November, we have evaluated over 1,900 gene-disease relationships in over 1,500 unique genes. And while most of these have been confirmed, there are several hundred in the limited and lower categories down here that may not be appropriate for clinical testing. So gene disease validity assessments first became publicly available on our website in 2016. And currently we have 47 approved gene curation expert panels, or GCEPs, across 15 clinical domains, and almost 1,000 people work on one or more GCEPs. And you will notice, of course, that GCEPs and VCEPs, which we'll discuss in a moment, have much larger workforces than either Dosage or Actionability. And this, of course, has a major impact on how quickly gene and variant curation work has been able to grow. So in the figure on the right, the number of GCEPs is illustrated by the blue bar, and the number of published curations is illustrated by the orange line. On average, the number of curations increases by 102% per year. And in 2022, we were able to complete an average of 35 new curations per month. Some notes in gene curation history. We first started work on developing the metric in 2013 and took several years to pilot and finalize the system. The first GCEP started up in 2016 and provided early testing for the framework, which was published in 2017. By 2018, those initial GCEPs had worked through their gene list, and we began to see publications describing their work. Also in 2018, ClinGen and a number of other partner organizations formed the Gene Curation Coalition, or GenCC, to support standardization in the gene disease validity evaluation process and to promote public data sharing. In 2020, ACMG recommended that ClinGen gene curation information be used when designing diagnostic gene sequencing panels, suggesting that only those genes with classifications of moderate or higher be routinely included on panels. And recently, in the past year, our guidance on how to determine whether disease entities should be lumped together or split apart for curation purposes was published. For variant pathogenicity, our curation question is, which variants within a given gene cause disease? Now, there are, of course, professional guidelines in place for interpreting germline sequence variants, but there are some cases where gene or disease level nuances need to be accounted for in order to provide more accurate interpretation. At this point, ClinGen has over 50 different variant curation expert panels, or VCEPs, working on such gene and disease level specifications. And as these are completed and approved, they're made available for public use. The VCEPs then use those specifications to classify difficult variants and to resolve discrepancies in ClinVar. In 2018, ClinGen received FDA recognition as a public human genetic variant database, and all curations from ClinGen VCEPs are designated as such in ClinVar. And at this point, ClinGen has generated over 4,300 variant classifications. So ClinGen VCEP classifications have been available in ClinVar since 2015. Currently, there are 22 fully approved VCEPs with a total of 55 in various other stages of development. And over 1,300 people work on one or more VCEPs. Here again, the total number of VCEPs, again at all stages, not just the ones that are completely approved, are shown using the blue bars, and the number of curations are shown in orange. The number of approved curation increases an average about 105% per year. And in 2022, we completed an average of 126 new curations per month. Again, going with our theme, just a few key moments in the variant curation timeline. In 2015, ACMG and AMP published the current sequence variant interpretation guidelines, Richards et al. And almost immediately thereafter, the first ClinGen VCEPs were formed. The goal of the VCEPs was, of course, to develop gene and disease-specific modifications to those general guidelines. And the first VCEP variant using said guidelines was actually submitted to ClinVar in late 2015. ClinGen developed its Sequence Variant Interpretation Group, or SVI, to provide general recommendations for how Richards et al. Might be used or modified. And their first recommendation came in 2017, where they described how to uniformly name ACMG AMP criteria modifications. Through the next year, they published several more recommendations, including those on the BA1, PBS1, and reputable source criteria, as well as the initial adaptations of Richards et al. Into a Bayesian framework. Around the same time, in 2018, the first VSEP manuscripts describing their rule modification and pilot processes were accepted. But most notably in 2018, that was the year that ClinGen became the first entity recognized by the FDA as a valid source of accurate human variant interpretation data. By 2021, we had published over 3,000 classifications to both ClinVar and the ClinGen evidence repository. And next year in 2023, we anticipate the release of updated sequence variant interpretation guidelines, a collaboration between ACMG, AMP, CAP, and ClinGen. So ClinGen is also branching out into other curation activities, including somatic cancer. The somatic cancer CDWG has over 160 members from 30 different organizations, including academic medical centers, laboratory diagnostic test developers, government agencies, and other partner consortiums. Their curation activities take place within 3 disease task forces focused on pediatric cancer, hematological cancers, and genitourinary cancers. And there are currently 6 somatic cancer VSEPs in various stages of development. These groups have been curating within the CIVIC database with plans to ultimately submit this information to ClinVar. Relatively recently, as the result of the collaborative relationship between ClinGen, CPIC, and PharmGKB, we've been able to display pharmacogenomic curation information on our website. And the summary of what's available is shown here. We do plan to continue our collaborations in this space and expand our scope. To this end, 2 new subgroups have been formed and are actively meeting. So we have a group focused on nomenclature survey development. This will be a collaboration with the Actionability Working Group to assess the needs of the larger PGX community for standardization and framework development. And we also have the Gene Drug Clinical Validity Framework subgroup. So this is focused on developing a gene drug response clinical validity framework that's consistent with our gene disease validity infrastructure. In addition to all of that, pharmacogenomics working group members are participating in other VSEPs and GSEPs to expand pharmacogenomics representation across clinical domains. So back to our infographic. Once we've used the aggregated data to answer the critical curation questions, we need to disseminate the information back to the scientific community. So we do this in a number of ways. In terms of our curations, one of the primary ways in which we make this information available is through our website. So of course, as you all are aware, curation results are made freely and publicly available shortly after completion. Our policy is not to hold them for publication, and we have no restrictions on their use. Our website is, of course, clinicalgenome.org. That's the primary portal for browsing curation data. However, variant curation data is also available in ClinVar and the ClinGen evidence data repository, and somatic cancer curation data is available in CIVIC. And over the years, our curation data has been utilized and displayed by numerous downstream users. So for example, clinical and research variant repositories like ClinVar or Decipher, resources describing gene disease relationships, so OMIM, GenCC, genome browsers like UCSC and Ensemble, variant interpretation tools and platforms like MuteSpliceDB or other commercial tools, somatic cancer variant resources like CIVIC, clinical decision-making resources like ACMG ACT sheets. And in addition, resources also use the unique canonical variant identifiers we talked about earlier from the allele registry, including ClinVar, Ensemble, Nomad, CIVIC, LitVar, and others. So clinicalgenome.org first became available in late 2014. And since that time, we've seen incredible growth, really closely corresponding with our increase in available curation data. So in terms of visits, the number of times people actually come to our sites, we have had over 914,000 visits thus far in 2022, with still a few more weeks to go. And our average growth is about 54% increase per year. In terms of our users, we have had 237,000 users thus far in 2022. That represents an average growth of about 31% per year. And over the past year, users have come from 194 different countries, 31.6% coming from the US and 68.4% from other countries. And once people get to our website, how many pages are they looking at? Well, we've had 3.3 million page views thus far in 2022, an average growth of 59% per year. Again, our users come from all over the world, and while the largest single group is from the United States, non-US users together make up the majority of the user base. And here you can see our top 20 user countries and their percent breakdowns. So we also disseminate information via educational events. We've hosted numerous such events over the years, but here on this slide, I'm just highlighting a few that took place in 2022. As you can see, they're a mix of in-person, virtual, and hybrid events, each with different areas of focus and different audiences. These events were well attended by individuals from around the world, and their virtual nature allowed for some of them to be recorded and viewed by many others at their leisure. In particular, I'd like to highlight our recent Curating the Clinical Genome meeting, which is our long-standing joint meeting with the Decipher team. This event was held in June, and it had over 470 registrants from 42 countries. We have several events already planned into 2023. Our next major event will be a workshop hosted by our data platform team at the Pacific Symposium on Biocomputing in January, followed by an invited session at the International Congress of Human Genetics meeting in Cape Town, South Africa in February. This year, the Decipher team will be hosting the Curating the Clinical Genome meeting in Kingston, and that will occur in July. Another notable method of disseminating information is through publications. At this point, we have about 100 publications or preprints describing consortium work, which have collectively been cited over 7,500 times. And finally, one of our most effective ways of dissemination is by engaging members of the community, such as yourselves, to participate in ClinGen curation activities. Through these efforts, people are able to learn more about our standards and their application, and then they're able to bring that information back to their personal practices and communities. The ClinGen Community Curation Program, or C3, was launched in October of 2018. And since that time, over 1,200 people from 75 different countries have taken our volunteer interest survey. Of those indicating interest, 754 went on to complete training. When we asked people why they volunteer with ClinGen, they endorsed a variety of reasons, but the most common of which was a desire to participate in the project or to gain curation experience or build their resume. Others indicated more altruistic reasons, such as promoting precision medicine standards, contributing to patient care, and helping the community. As Jenny mentioned earlier, just as an incentive to our volunteers and others, we offer several different ways of obtaining continuing education credit by participating in our activities. And to that end, we've successfully set up modules for each of our major curation activities available through the ACMG Genetics Academy. So with that, I would just like to acknowledge the fact that none of what I described to you today would be possible without you. At this point, we have 2,142 individuals around the world who devote their time and their resources to ClinGen and creating this resource for our community. And we absolutely could not do it without each and every one of you. So huge thank you to you. And with that, we can take any questions.
Thank you so much, Erin. It really is amazing to see how much progress has been made, and that was a wonderful review, so thank you. Any questions for Erin? I see there's a note in the chat. Oh, I think this just came to me. Might be useful to mention that somatic variance information is also available in ClinVar.
Right.
And that the ClinGen CGC VICC standards for the classification of pathogenicity of somatic variants in cancer publication is available.
Wonderful. And do you want to put that in the chat? I assume that is Jason putting that information in just so people can access it, the publication?
Yep, I will put that in. There we go.
Perfect. Everybody check that out. And if anybody has anything else, I know there are a number of other ClinGen staff members on the line. If anyone, obviously you can't there, and that's not even all of it. We even have more stuff that I just couldn't fit in there. So if anybody wants to take a moment to mention anything that I didn't highlight, we do have time, so feel free.
Yep, I do see another note in the chat says this is a really nice review of the history and intent of ClinGen, and is it okay to share the recording of this meeting with individuals who are considering a group?
That's fine with me, if that's okay with you, Jen.
Yeah, perfect. And I will be sharing the link via the listserv by tomorrow at the latest, and I will also put the link on our Biocuration educational materials webpage, so it will be available there too, but feel free to share that as needed. And then Erin, there's also the general email for ClinGen as well, right? If people had any particular questions that if they weren't sure who to address those to, there's the general ClinGen email.
It's right here, and that email will come to me. So feel free to send it.
Okay,